Hello everybody. So now that you know about transcription and translation, there are two final topics I want to talk to you about. The first is mutation, and the second is the processing of the mRNA transcript. So let's get started. During our unit on evolution, we saw that mutation is one of the major causes of variation in organisms, and that without variation in a population of organisms, evolution itself would not be possible. A mutation occurs when the base sequence of the DNA in the gene changes, and this often can occur during replication. Mutations can occur when parts of a chromosome move from one to another, or when a piece of a chromosome gets duplicated, but much more common are what are called point mutations, and point mutations are simply a change in one or a few bases in the chromosome. There are basically two types of point mutations, what are called substitutions or insertions and deletions. We're going to look at each one of these individually. A substitution occurs when one nucleotide and its partner is replaced by another pair of nucleotides. Some point mutations are silent, and what that means is they do not change the amino acid in the polypeptide. So you saw that there was repetition in the genetic code, and it would be possible that when a mutation occurs, that the codon in the mRNA is changed only slightly, but the amino acid it produces during translation is not changed at all. Some point mutations may change one or a few amino acids, but have no effect on the function of a protein. And these are also de facto silent mutations because they can't be seen and they don't have an effect on the functionality of the protein. Some amino acid changes must occasionally improve the protein, and such a change will be selected for by natural selection because it's an improvement. It increases the fitness of the individual that has the mutation. And some mutations must produce a non-functional or a less functional protein, and therefore will be selected against by natural selection. So that's the way to think about the effect of mutations. They can be silent because they don't change the amino acid or they don't change the functionality of the protein, or they can improve the protein, or they can make the protein non-functional or less effective. It turns out that improving the protein is the least common, and in fact probably pretty rare, whereas silent and less effective mutations, what are called deleterious mutations, are much more common. In addition, we can think about the mutations as causing two different kinds of effects. Base pair substitutions that code for an amino acid but not the same amino acid are known as missense mutations. Whereas when a changed base pair results in an early stop codon, this is called a nonsense mutation. And a nonsense mutation nearly always causes a non-functional protein to be produced, whereas missense mutations often have little or no effect on the protein itself, although in some cases they can have a tremendous effect. Let's look at this in pictures now. Here we have the original mRNA and the protein that gets produced by it. And now we have a mutation where instead of it being GGC in the fourth codon, it's GGU. But GGU codes for glycine just as GGC does. So this has no effect on the amino acid sequence, and it is a silent mutation. Here we have an A in the first position of the fourth codon instead of a GE, and this causes a new amino acid to be put in at the end, serine in place of glycine. That is a missense mutation. In the third example, we have the second codon where there was formerly an A, there is now a U placed. This gives us the codon UAG, which is a stop codon. That means that all these downstream codons are never translated. And this is called nonsense. So in this case, the protein would be missing its last three amino acids. Let's take a look at insertions and deletions. Insertions and deletions are additions and losses, respectively, of nucleotides in a gene. When an insertion or deletion occurs, it alters what's called the reading frame, or you can think of it as the triplet grouping of the mRNA, and therefore produces what is called a frame shift mutation. Now, if the number of nucleotides inserted or deleted is a multiple of 3, then only the codons that are inserted and the ones surrounding it can be affected. So when there are multiples of 3 in an insertion and deletion, only some of the codons are affected. But if they're not a multiple of 3, then all the downstream nucleotides will be improperly grouped into codons, and it will result in extensive missense and maybe even nonsense. The protein in this case has a high probability of not being functional because of the tremendous number of amino acids that will be incorrectly placed. Let's take a look at this. Here we have an insertion of a U before the AAG codon up here. That now produces the UAA codon, which is a stop. So we say that frame shift causes immediate nonsense. Nonsense, remember, is an early stop codon. In the second example, there is a deletion in the UUU codon, so that codon now becomes UUG. That changes the amino acid from phenylalanine to leucine. But now look what happens downstream. Instead of having the GGC codon for glycine, we get the GCU codon for alanine. So now this one is incorrect. And then there are going to be others incorrect. The stop that was supposed to be here is going to be missed, so on and so forth. And so you will have a bunch of amino acids that don't belong there. In this case, frame shift causes extensive missense. Finally, look what happens when we delete 3 nucleotides. In this case, we took out the AAG, the second codon. That's removed. And so what happens now is the sequence of amino acids, instead of being methionine, lysine, phenylalanine, glycine, is methionine, phenylalanine, glycine. This may have a big effect on the protein, but the loss of one amino acid may not significantly affect the protein that's produced. So in this case, there is no frame shift, but there is a missing amino acid in this case. If there had been an insertion, there would be an extra amino acid. So that is an overview of mutation. We will do some worksheets in class where you will get to apply these ideas and solidify them in your mind. The second topic today is RNA processing. In eukaryotes, after the mRNA is made, it has to be processed, or what's called sometimes edited, to produce a finished RNA. It turns out that within a gene, there are regions that are expressed, which means they're translated as polypeptides, and other regions that will never be translated. And now here's some vocabulary. Regions that are translated are called exons, and regions that are not translated are called introns. And you can think of the exons as the expressed regions, and you can think of the introns as the intervening regions. Here is what a gene looks like. We have the DNA, it has a promoter region. This is the region that the RNA polymerase is going to bind to, and then the mRNA transcript is going to be made by reading the whole line. But notice there are both exons and introns here. Notice also that the final mRNA is only composed of exons, the blue sections. The other regions have been excised or cut out, right? The promoter region and all the introns are cut out. The introns are removed and recycled, and the exons are then spliced back together to produce the final RNA. The original RNA is sometimes referred to as the pre-mRNA, and I think that was in an earlier diagram in one of these videos. At this point, you're probably asking, why the heck do introns exist? And that's a fantastic question. Why would there be regions of so-called junk DNA within a gene? It turns out that our chromosome contains about 25,000 genes, but our body itself has the ability to make about 100,000 different proteins. So as you can see, that means that on average, each gene must be able to make 4 different proteins. So how does this happen? It is now believed that the exons code for polypeptides that have specific functionality. In other words, they add a certain chemical element to the protein they're going to make. And so different proteins can be made by mixing and matching different exons. For example, on one gene with 10 exons, a certain protein might be made by using exons 1, 2, 4, 5, 6, 7, 9, while a different protein might be made by using exons 2, 3, 4, 6, 8, 9, and 10. And those would be two different proteins, and they would have two different functions. The editing and splicing of these exons is done by a separate class of RNA molecules that we're not going to study in this class, but that you will study in an advanced biology course. Finally, one of the most amazing things about the final mRNA is it has a 5' cap, and I'm not going to talk about that, but it also has what's called the poly A tails. A bunch of A ribonucleotides are put on at the end. So why does the back end of the mRNA have a long sequence of A nucleotides? Once the mRNA is translated, after it has been used, it needs to be recycled. And that job, of course, is done by an enzyme that removes nucleotides. And like many of the DNA and RNA-related nucleotides, it can only do so starting at the 3' end. You'll notice the poly A tail is at the 3' end. Once the mRNA has reached the cytoplasm, this enzyme will find it and begin taking it apart. So the poly A tail has evolved as a protective measure for the mRNA transcript against this recycler enzyme. And that's about all you need to know about transcription and translation. Today, what I hope you have learned is the details of what a mutation is. Mutations can involve full chromosomal rearrangements, but the only ones we're going to study in this course are point mutations, where only one or a few nucleotides are affected. Point mutations can either be a base pair substitution, where one base substitutes for another, or they can be insertions and deletions, where extra bases are added or deleted. Once a mutation occurs, if it is a substitution, it can be a silent mutation, which means it has no effect on the protein. It can improve the protein, which is a very rare outcome, or it can make a protein that doesn't function well, in which case we call it a deleterious mutation. And we can also talk about the mutations as causing missense when they change the amino acid, or nonsense when they put an early stop codon into the mRNA transcript. Insertions and deletions, if they are not multiples of 3, cause extensive frame shift and extensive missense and often nonsense. The other thing we learned today is about the processing of mRNA. After the mRNA is made, the introns need to be cut out, the exons need to be arranged into a functional mRNA transcript. That mRNA transcript will travel to the cytoplasm, where it will get attacked by a recycler enzyme. And if that happens before translation happens, then you will produce a non-functional protein. As a result, cells have evolved to put a long poly A tail, a sequence of AAAAA nucleotides at the 3' end of the mRNA, so that the recycler enzymes never destroy the polypeptide message. So that's it for today. If you have time, go outside, take a look at nature, ask biological questions, bring them back to class, and I'll see you soon.